Guess who's back? Still doing this, Andre? Oh, for sure. Yeah.
Okay, we're looking at cAMP signaling, CAMP signaling. So let's just look at this image for a while. We're looking at the steps 1 through 5. Sorry, 1 through 6. Okay, so let's just talk about this a bit while we're looking at the picture. CAMP signaling pathway. So remember that we're starting with a signaling molecule or ligand, and then we're hoping to stimulate the production of a protein. So this is the end point. So let's talk about the levels. So first is number 1. We can see the signaling molecule or ligand coming in and attaching to the receptor. So this is number 1 is the ligand binds to the cell receptor. Number 2, let's look at adenylyl cyclase. Adenylyl cyclase is activated by the guanosine triphosphate GTP bound G protein subunit A. It's separated from the beta and gamma subunits, and then cAMP is formed from ATP. So that's what's happening in 2. So now let's follow the CAMP and see what happens. CAMP, the second messenger, then binds to the regulatory subunits of the CAMP-dependent protein kinase A, and releases the catalytic subunits. So now we're moving downstream, and things are happening within the cell. Number 4, CAMP is degraded by cAMP-dependent phosphodiesterase because it's no longer needed. And then number 5, the activated catalytic subunit translocates into the nucleus and phosphorylates the transcription factor CREB, which is the CRE binding protein bound to the CAMP response element CRE. And then we get the specific gene expression of inducible genes. So this is what occurs finally. So remember the beginning, the middle, and the end. What is the CAMP signaling pathway, or the second messenger pathway?
It's a whole nother day. Come back a baby, Snoop Eagle Level G. I represent, been away for a while, but you all know I'm still doggy style, no death row.
Okay, let's look at this slide.
It's a sticker. Get up. They got your risk on the shelf for mine too. And guess who they gonna pick up? Some fools think they know a hit, but they don't. Some inmates escape.
Okay, let's take a look at the slide, and we're looking at this basic process again, ATP to CAMP or cyclic AMP. So how does this happen? Well, you can see by looking at the chemistry here. Very simply, at the top you have ATP, and then it becomes CAMP. So 2 phosphates are lost somewhere. If you look closely, you can see that starting at the top, ATP is activated by G protein, and activated by adenylyl cyclase. So going along down the line, this adenylyl cyclase is activated, and as it is activated, it loses 2 oxygens and 1 hydrogen. So this is a hydrolysis reaction, and bonds are formed between oxygen and carbon, and then this bond is broken, and going on down the line you see that finally CAMP is deactivated, by a phosphodiesterase. And this is the deactivation step. So what's happening here is that because the cAMP cannot be left running around stimulating things, we have to get rid of it, similar to what happens with phosphorylated proteins. So it's analogous to the kinase phosphatase reactions, but here the cAMP is sort of recycled, or it's sent back deactivated. Let's take a look at this for a second. So remember here, the main parts of the G protein is working on adenylyl cyclase to change ATP to cyclic AMP. Then the phosphodiesterase changes it back to cAMP. And in this case, it's 5' AMP, so it's no longer cyclic. So it can no longer be used as a signaling molecule. So it is the same basically as the top molecule, except that it's missing 2 phosphates. So now we know how ATP becomes cAMP, and that adenylyl cyclase is involved in this situation, and it is a hydrolysis reaction. So let's understand the metabolism of cyclic AMP. The main part of this slide here, the main point here is we're looking at signal amplification, and we should understand that this is not a 1-to-1 ratio. The second messengers, you are actually multiplying your ratio here, that once the receptor binds to the G protein complex, it's more than one G protein complex, so you're having a lot of things happening in the cell. So what's happening is each activated receptor complex can stimulate multiple G proteins, and each activated adenyl cyclase can then generate many cyclic AMP molecules. And this E here is the enzyme activity, in this case protein kinase A. And then we have phosphorylase is another enzyme, and we're ending up with the glucose. But it's important that we understand this process, that what is used to stimulate the activity and how we get there. Anyway, that was just a short introduction to signal amplification using G proteins, adenyl cyclase, and cAMP.